Our concept of the gene has undergone a remarkable evolution as biologists have learned more and more about the nature of inheritance. The earliest studies revealed that genes to be discrete factors that were retained throughout the life of an organism and then passed on to each of its progeny. Over the following century, these hereditary factors were shown to reside on the chromosomes and to consist of DNA, a macromolecule with extraordinary properties. In this lecture, we take a closer look at the molecular biology of the DNA and its replication. We also consider how the knowledge of DNA structure and replication has been used in the development of the laboratory techniques for isolating fragments that contains genes or parts of genes of particular interests, and for determining the sequence of bases in the DNA fragments. Before we go into the deeper concepts of DNA structure, replication, and manipulation, let us ponder these terms. First, we have the genes. Genes are fragment or the section of DNA that codes for a protein or for the messenger RNA. And when we say DNA, it is a double-stranded molecule of a polymerized nucleotides, or this is a deoxyribonucleic acid. The nucleotides are composed of ribose sugar and a phosphate group and a nitrogenous bases. And a nucleic acid are made up of DNA and RNAs. Further, a ribose sugar is a 5-carbon sugar. This illustration is a double-stranded DNA in a right-handed helix of paired complementary antiparallel structure. Observe that the nucleotide bearing the adenine, the cytosine, guanine, and thymine, which Watson and Crick based pairing between the adenine and the thymine, the guanine and the cytosine, which is in the other complementary strands that helps hold the strands together. The complementary is also the key to replication because each strand can serve as a template for the synthesis of the new one. The genome differ tremendously even among the closely related organisms. Genome is a genetic complement of a cell or a virus. In eukaryotes, the term is commonly used to refer to one complete haploid set of chromosomes that are found in egg and the sperm cells. Notice in the table that even these are group of virus, they differ in approximate genome size in thousands of nucleotides. Also, in this group of eukaryotes here, we can observe that the differences of their genome size in thousands of nucleotides. The units of length of nucleotides or nucleic acid in which genome sizes are typically expressed into kilobase, which is 10 raised to 3 nucleotide pairs, or the double-stranded, or 10 raised to 3 nucleotides for the single strand. In the megabase, or MB, it's 10 raised to 6 nucleotide pairs, or 10 raised to 6 nucleotides for the single strand. The DNA is a linear polymer of 4 deoxyribonucleotides. So DNA is a polymer, as what I've said, that is a large molecule that contains repeating units and is composed of 2' deoxyribose that means with 5 carbon sugar, phosphoric acid, and 4 nitrogen containing base, or these are adenine, thymine, guanine, and cytosine. So let's take a look into the components of nucleotides. First, we have the sugar. So this is the typical structure of a sugar of the nucleotide. As what I've said, this is a ribose sugar that contains 5 carbon. So we start numbering the carbon of the ribose sugar starting here, 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. Notice in our sugar, we have 2 OH group attached to 3 prime and 5 prime of the ribose sugar. And on top is the oxygen. Notice also in the 2 prime carbon of the ribose sugar, there is an X. How do we know if this sugar is deoxyribose or a ribose sugar? By substituting H or OH in this X. If X is H, therefore this is a DNA, that means a deoxyribose sugar. But if X is OH, or the hydroxyl group is attached to your carbon number 2 or 2 prime carbon of the ribose sugar, this is said to be RNA or ribose. Another component of nucleotide is its nitrogenous base. And we have 2 types of nitrogenous bases. These are purines and pyrimidines. When we say purines, these are double ring structure, and these are adenine and guanine. And when we say pyrimidines, these are nitrogenous base that have a single ring structure, and these are cytosine and thymine. For the general formula of a pyrimidine, we use this. And notice that a pyrimidine has a single ring. Notice also in this general formula of a pyrimidine is the X here and the Y. The same in our previous slide, we used to substitute these groups into the X and Y. So if X is NH2, therefore it will be the cytosine. And if X is oxygen, it will become uracil. And if X is O or oxygen and Y is methyl or the CH3, therefore the pyrimidine is said to be thymine. Remember always that if oxygen, it is always double bond. The IUPAC name for a cytosine is 2-oxo-4-aminopyrimidine, and for uracil, it's 2,4-dioxopyrimidine. For the thymine, it is also known as 2,4-dioxo-5-methylpyrimidine or 5-methyl-uracil. Next type of nitrogenous base is a purine. So this is the general formula of a purine. Notice that purine has 2 rings. So again, notice that we have X here and Y here. Again, we will follow if X is NH2 or amine group, then therefore this will be an adenine. If X is O, still it's adenine. But if Y is NH2, it is classified as a guanine. For us to remember what are the nitrogenous bases that belongs to a pyrimidine or into a purine, let us remember for purine, it's adenine and guanine. So we use the term PUAG, purine, adenine, guanine. For pyrimidine, we use the term PICUT, P-Y-C-U-T, PICUT. So PICUT, that means pyrimidine, cytosine, uracil, and thymine. Again, remember PICUT and PUAG. PICUT, pyrimidine, cytosine, uracil, thymine. PUAG, purine, adenine, and guanine. Another component of a nucleotide is the phosphate group. The phosphate group usually imparts negative charge to the DNA. So this is the general structure of our nucleotide that is composed of a nitrogenous base, a sugar, and a phosphate group. So in the case of DNA, the sugar is said to be deoxyribose, and for RNA, the sugar is ribose. And notice that the bond between the ribose sugar or deoxyribose sugar connected to the nitrogenous base is known as your N-glycosidic bond. In DNA, each base is chemically linked to one molecule of sugar deoxyribose, forming a compound called the nucleoside. When a phosphate is attached to the sugar, the nucleoside becomes nucleotide. In this illustration, we can observe that in each base, the nitrogen is linked to the deoxyribose sugar, which is indicated. Atoms with darker backgrounds participate in hydrogen bonding between the DNA base pairs. Again, notice that these nitrogens here are linked to our deoxyribose sugar, while these atoms here with darker backgrounds participate in hydrogen bonding between the DNA base pairs, such as the link between a guanine to cytosine. Note, in conventional way, the atom in a sugar are given a prime numbers to distinguish them from atoms in the bases. So the base is attached to the 1' carbon, and in nucleic acid, the nucleotides are joined to form a polynucleotide chain in which the phosphate attached to the 5' carbon of the sugar is linked to the hydroxyl group attached to the 3' carbon of the next sugar line. Notice also in this illustration the chemical bonds by which the sugar components of adjacent nucleotides are linked together through phosphate groups that are called as phosphodiester bonds. And the 5' end, which terminates in a phosphate, whereas the other end here is in 3', which terminates in a hydroxyl group. Another distinguishing structure of a DNA is that duplex, that means a double helix in which the bases form a hydrogen bonds. The duplex molecule of the DNA consists of 2 polynucleotide chains twisted around one another, forming a right-handed helix. Each chain makes a complete turn every 34 Armstrong, and the bases are spaced at 3.4 Armstrong. Thus, the 10 bases per helical turn in each strand, or 10 base pairs per turn of the double helix. And to stabilize the double helix structure of the DNA, each base pairs are bonded by what we call a hydrogen bond. A hydrogen bond is a wick in which 2 negatively charged atoms shared a hydrogen atom. And hydrogen bond contribute in holding the 2 strands of the DNA together. Paired bases are said to be planar, parallel to one another, and perpendicular to the long axis of the double helix. In this illustration, the adenine and thymine has 2 hydrogen bonds, and the guanine and cytosine has 3 hydrogen bonds that stabilize the double-stranded DNA. That means the most stable connection or linkage between the nitrogenous bases of the 2 strands of DNA is the connection between guanine and cytosine. Since this bond is stronger between the G and the C, or the guanine and cytosine, it requires more energy to break the bonds. Also note that the specificity of base pairing means the sequence of bases along on the polynucleotide strand of the DNA is matched or complementary with the base sequence in the other strand. However, the base pairs along a DNA can be arranged in any order, and the sequence of bases differs from one part of the molecule to another, from species to species. No restriction on base sequence. The DNA has virtually unlimited capability to code a variety of different protein molecules. In a DNA, we have the backbone, and the backbone of each polynucleotide strand consists of deoxyribose sugar and alternating phosphate groups. When we say deoxyribose sugar, these are sugars that are attached with phosphate groups by a phosphodiester bonds that links the 3' carbon atom of sugar to 5' carbon of the next sugar next in line. The 2 strands of polynucleotide will run in opposite directions and are said to be anti-parallel. So 2 grooves spiraling along outside of the helix, that proteins may interact, or we should say contact, during the replication. And these are known as our minor groove and the major groove. In this illustration, notice a segment of a DNA molecule showing anti-parallel orientation of the complementary strands. Arrows actually indicates the 5' end up to the 3' end of the strand. On this portion of the strand, it starts with 3' end up to the 5' end. So the phosphate groups here in both sides join at the 3' carbon atom of 1-deoxyribose to the 5' carbon atom of the adjacent deoxyribose. Again, the phosphate group here is joined between the 5' carbon and into the 3' carbon of the deoxyribose sugar. The same also in this parallel strand. The nucleotides are said to be added one at a time to the growing end of the DNA strand. So primary function is to produce the base sequence of the parent molecule, and the specificity of base pairing provides the mechanism used by all genetic replication system. In the nucleotide monomers, usually are added one by one to the end of a growing strand by an enzyme called the DNA polymerase. So the sequences of base in each newly replicated strand or a daughter strand is complementary to the base sequence in an old strand or parental strand being replicated. For example, wherever an adenine nucleotide is present in parental strand, a thymine nucleotide will be added to the following end of the daughter strand. And the DNA replication is said to be semi-conservative. So that means the parental strands remains intact. So the parental strands here is still intact. Note that in each parental strands serves as a template for a new strand or the hydrogen bonded to its parental template. As replication proceeds, the parental double helix unwinds and then rewinds again into a double helices contains one original and one newly formed strand. So notice in this illustration that this one is a parental strand and another one is the parental strand. So they produce as they replicate or they produce undergo replication. Notice that there is an addition of the new strands. So here, the two identical molecules of the DNA result each composed of one parental strand and one new strand. And that is the reason why it is said that the DNA replication is semi-conservative, because it retains the parental strand intact. So before replication of the DNA strands, the DNA must be separated first, or should I say unwind first, by means of this helicase enzyme. Again, unwinding the double helix to separate the parental strand requires an enzyme known as a helicase enzyme. It usually hydrolyzes ATP to drive the unwinding reaction. Without the presence of this helicase enzyme, the parental strand cannot be separated and cannot produce new strand, and it cannot replicate. Note also that most cells have several helicases specialized for different roles such as replication and recombination or repair. Further, once the unwound the strands would tend to come together again so that they must be stabilized. And stabilization of unwounded strand is the work of the SSB, or the single stranded DNA binding protein. And in the illustration, here is the SSB proteins. So again, SSB proteins bind and stabilize single-stranded DNA at the replication fork. SSB proteins, it has an affinity for single-stranded DNA at least 1,000-fold greater than that for double-stranded DNA. In the case of Escherichia coli or the E. coli, apparently the same SSB protein is used in the DNA replication, recombination, and repair. Notice also this gyrase, or known as the topoisomerase 2. This gyrase usually cleaves and swivels DUPLEX to release mechanical stress of the unwinding DNA strand. In replication, each new DNA strand or fragment is initiated by a short RNA primer. So, DNA polymerase is unable to initiate the synthesis of a new strand. It can only elongate an existing strand at the 3' end. So, it is initiated by a special type of RNA polymerase, and this RNA is usually a single-stranded nucleic acid consisting of 4 types of nucleotides joined together by the 3' to 5' phosphodiester bonds with the same into our DNA. So how do we distinguish RNA from DNA? First, we can distinguish a difference between RNA from a DNA through its sugar component. So RNA contains ribose, which is identical to the deoxyribose of the DNA, except for the presence of OH group, or the hydroxyl group, on the 2' carbon atom. Also in their nitrogenous bases. Usually, thymine found in the DNA is replaced by the closely related pyrimidine, which is uracil in the RNA. In RNA synthesis, a DNA strand is used as a template to form a complementary strand in which the bases in DNA are paired with those in the RNA. And it usually catalyzed an enzyme called the RNA polymerase. The RNA polymerase can initiate the synthesis of RNA without the needing of a primer, while DNA synthesis is initiated by a short stretch of primer RNA. As what I have mentioned earlier, DNA synthesis needs a primer. So the short stretch of RNA is shown and the primer is later removed. In the first step, the DNA polymerase adds to a deoxyribonucleotides to the 3' end of the primer. So where is the 3' end of the primer? Here in this portion. And the DNA polymerase has a proofreading function that usually corrects errors in replication. There will be a big problem if there is a wrong template when the DNA replication occurs, because it will signify later on a change in the base pair and mutation to the organism. Thus, DNA polymerase is very important function for proofreading of the codes. The enzyme DNA polymerase forms the sugar phosphate bond or the phosphodiester bond between adjacent nucleotides in a new DNA acid chain. So the reaction is catalyzed by the DNA polymerase, which is the formation of phosphodiester bond between the 3' hydroxyl group of the chain being extended and the innermost phosphorus atom of the nucleoside triphosphate being incorporated at the 3' end. In the 3' hydroxyl group at the 3' terminus of the growing strand attacks the innermost phosphate of the incoming nucleotide and forms a phosphodiester bond, releasing the 2 outermost phosphates. In this illustration shows that the recognition step is shown as the formation of hydrogen bonds between the adenine and thymine. So the chemical reaction occurs when the 3' OH group of the 3' end of the growing chain attacks the innermost phosphate group of the incoming trinucleotide. So let's say here in this portion, so the base pairing specifies the next nucleotide to be added at the 3' end. So to make it larger, this one here, the 3' hydroxyl group at the end of the 3' or the 3' end of the growing strand attacks the innermost phosphate of the incoming trinucleotide, causing, of course, the O and P bond is formed because hydrogen is released already and forming new nucleotide. So that's why the 2-phosphate group is released in the cell. So this one will left already forming a phosphodiester bond. The DNA polymerase 1 and the DNA polymerase 3 are the two DNA polymerases that are needed for the replication of the Escherichia coli. So the DNA polymerase 1 plays an essential yet secondary role in the replication, while in the DNA polymerase 3, it is a major replication enzyme of the E. coli. So exists in the cell as a large protein complex that is responsible for the elongation and the replication of DNA at origins of replication and the addition of deoxynucleotides to the RNA primer. Eukaryotic cells, on the other hand, contain several DNA polymerase, and we call it polymerase delta. In mitochondria, have their own DNA polymerase to replicate the mitochondrial DNA. Notice before that our mitochondria can reproduce on their own because they have their own DNA. Thus, they have also their DNA polymerases for replication. Major DNA polymerases also have an exonucleases that can break the phosphodiester bonds in sugar and phosphate backbones of the nucleic acid chains. Your exonucleases provides a built-in mechanism for correcting rare errors in the polymerization. By the exonucleases activity of DNA polymerase is often called as editing function or proofreading function. So the proofreading function can look back only one base. So meaning the one added last. So the genetic significance of proofreading is that it is an error correcting mechanism that serves to reduce the frequency of mutation resulting from incorporation of incorrect nucleotides in the DNA replication. We all know that mutation may cause diseases, for instance, and some other malformations in our body. In this last slide, we have the dideoxynucleoside analogs are also used in the treatment of diseases. With our knowledge about the DNA structure and replication, it leads to the development of drugs for clinical use such as treatments for AIDS. But unfortunately, we are ongoing study with the antiretroviral drugs for treating AIDS. Recall that the nucleoside is a base attached to a sugar without the phosphate. So the nucleoside analog is a molecule similar but not identical in the structure to a nucleoside. Presently, the DDC is the normal dideoxyribocytidine nucleoside. So it is actually a effective chemical against AIDS. Like such, for example, we have the AZT, the D4T, and the DDI, and other such analogs. When we say AZT, it is a 3-azido-2,3-dideoxythymidine. The DDC here is 2,3-dideoxycytidine. The D4T is 2,3-dihydro-2,3-dideoxythymidine, and the DDI is 2,3-dideoxyinosine. So to conclude, these analogs of DDC is usually effective in treating AIDS. But as of the moment, we don't have drugs that can treat or can kill the virus, but instead suppressing the viral load in the body or in the system of the patient with AIDS. This ends my presentation. Thank you for your kind attention. God bless and keep safe, everyone.